Okay, welcome to the second to last video for our module on DNA and RNA. So in this video, we're going to continue with our discussion of transferring information from DNA to RNA to a protein. We've talked about transcription, DNA to messenger RNA. Now we're going to discuss how you go from messenger RNA to a working protein. And that happens through the process of translation. So translation is where we convert different languages. So from the nucleic acid language to the protein language. And this requires more elaborate cellular machinery compared to that transcription process. Okay? But in order for translation to happen, you need to have that working messenger RNA molecule that's produced by transcription. Once that messenger RNA molecule is present, then your ribosomes, your transfer RNA, can work together with the messenger RNA to generate a working protein. And they will do this by using energy. So building proteins require energy in the form of ATP. And the translation from mRNA into amino acid also requires what we can classify as an interpreter. And that interpreter is known as transfer RNA or tRNA. So there are three kinds of RNA molecules. You have mRNA, tRNA, and rRNA. So mRNA, we've already discussed, is that messenger RNA. rRNA is the RNA molecules that make up ribosomes. And remember, ribosomes are made in the nucleus and then released into the cytoplasm. Your tRNA is used to match specific amino acids to their appropriate codons. So basically following that table we looked at a few videos ago. And in order for this to happen, the tRNA molecules have to do two different things. First, they have to pick up the appropriate amino acid. Second, they have to recognize the appropriate codons in the mRNA molecule. And so the unique structure of tRNA molecules enables them to form both of these individual tasks. All right, so with the tRNA molecule, that transfer RNA, like I mentioned earlier, it's made up of an RNA molecule itself. So it's actually a strand of approximately 80 nucleotides, which makes up that RNA molecule. And that tRNA molecule twists and folds on itself to form the transfer molecule that brings in the appropriate amino acid. So at one end of the transfer RNA, you have a special triplet of bases that's referred to as the anticodon. This anticodon is complementary to the codon triplet of the mRNA. So it follows those base pair rules. And so during the translation, the anticodon recognizes the codon of the mRNA and it brings in the associated amino acid and attaches it to another amino acid to build the growing polypeptide and protein strand. So here, for example, we have the 80 or so base pairs, right, that make up the tRNA molecule. Here is the anticodon that complements the codon on the messenger RNA. And this is the area where the amino acid attaches to that transfer RNA. So this is a simplified representation. You basically have the tRNA molecule with its anticodon and then the attachment site for the associated amino acid. Okay, so with translation so far, we said that you have to have messenger RNA, you have to have transfer RNA, And you also have to have a ribosome. The ribosomes are the organelles that coordinate the mRNA and the tRNA together and make those polypeptides. And like I mentioned a while ago, the ribosomes are made up of rRNA, so ribosomal RNA. And you get a fully assembled ribosome that has a binding site for both mRNA and tRNA. The binding site for mRNA is on what we call a small subunit, and the tRNA binding site is on what we call the large subunit. And the large subunit actually has two tRNA binding sites, one that's called the P site and one that's called the A site. So if we look at a figure of this, we have your small RNA subunit, your large RNA subunit, or I'm sorry, your ribosomal subunit and small ribosomal subunit. The mRNA binding site is basically where these large and small subunits kind of connect. The P site and A site exist on the large subunit, and that's where the tRNA binds. So basically what you have is you have a small subunit and a large subunit. You have mRNA, you have tRNA, and you have the associated amino acids. So your mRNA, your small subunit, and your large subunit will bind together, and then your tRNA will come in with the anticodon of the specific codon and bring in the associated amino acid. And this whole process is broken down into three phases, exactly the same name as they were in transcription. So translation has initiation, elongation, and termination. So initiation brings together the mRNA, the first amino acid that's attached to the tRNA, and your two subunits, the large subunit and the small subunit. Okay. The mRNA has the cap and tail. Remember, it was modified with that cap and tail that help it bind to the ribosome. So that cap and tail is very important for binding to that ribosome. So here's an example. You have cap, starting genetic message right here. You have the end, then you have the tail. Okay, so that's binding to that ribosomal subunit. So during initiation, the mRNA molecule binds to that small ribosomal subunit, and then a special initiator tRNA comes in and binds to that start codon. And if you remember, that start codon is AUG for methionine. And that's where the translation begins. So if the start codon is AUG, then the anticodon would be UAC, right? Because they would pair together. And so once initiation is complete, the amino acids are then added on one by one to the first amino acid, which is methionine. And so it basically has this three-step elongation process that's shown on the next slide. So basically initiation is the attachment of the small subunit, the mRNA, and the initiator tRNA with that starting codon. And then the large subunit comes in and binds, making the ribosome translation unit complete. So we have that small subunit, the mRNA, the initiator tRNA that has methionine. And see, you have AUG, which are the first three codons, or the first codon, and the complementary anticodon on the tRNA is UAC. Now this initiator starts off in the P site. Now what happens is the next codon will be read. So these next three, the next three nucleotides, and the tRNA molecule with the associated amino acid will come in to the A site and start elongation. And that's what happens here. So you have that P site, the next codon with the associated anticodon, tells the tRNA to bring in a specific amino acid. It binds into the A site. Then the growing polypeptide chain in the P site will attach to that A site amino acid. Then tRNA from the P site will be kicked out. The one in the A site will move over, and then you'll continue the process. So you'll continue elongating that polypeptide chain. And so elongation continues until you reach a stop codon. And so remember, there are those 3 stop codons. And once that stop codon reaches the A site, the completed polypeptide is released, and then the ribosome breaks back up into its individual subunits. Okay. And so just to review kind of this transcription translation process, so going from DNA to a protein, right? We have a figure showing that in the next slide. For eukaryotic cells, DNA to RNA, and then that RNA is modified into messenger RNA. And that has to be processed before that messenger RNA leaves into the cytoplasm, which is out of the nucleus. Once that process happens, translation occurs in the cytoplasm, either in free-floating ribosomes or ribosomes that are attached to the endoplasmic reticulum, where the associated polypeptides and proteins will be made. So you have this figure here. So you have transcription in the nucleus, which makes that RNA molecule. That RNA molecule is modified with the tail and the cap. The introns are cut out. The exons are spliced together. Then that messenger RNA will leave the nucleus out of the nuclear pore and into the cytoplasm, where it will then join with the ribosomal subunits and tRNA molecules in translation to build a polypeptide and to make a functioning protein. Okay, and so that's it for our information on transcription, translation, and basically how you go from DNA to RNA to a protein. The last video, that is only probably going to be a couple of minutes, is going to focus on mutations and modifications of DNA. So we'll see you in that final video for Module 10.